everyone. My name is Adijat Jimoh. I'm a PhD candidate, as Anna said, and thank you, Anna, for your supervision during this long PhD. So today I'll be sharing some insights from my PhD on the vaginal, viral, and bacterial ecosystem among African women. And I'll take you through a roadmap in my talk, structured around these 5 facets, starting off with what do we know about the vaginal microbiome in African women, and what exactly are we missing? What's the gap in there? And then this will next focus on what are the existing vaginal viral communities amongst African women and how diverse are they in terms of their genomes and so forth. This will lead me into talking about bacteriophages and how they possibly interact with vaginal bacteria, and also leading to a clinically urgent question as to if compounds that women are commonly exposed to in sub-Saharan Africa, if they can induce prophages within vaginal bacteria. And this would now pivot to a parallel arm of my work, looking at pregnancy, how the vaginal bacterial communities shift during pregnancy in women with and without HIV, and what are the possible contributions of antiretroviral therapy. And I'll end up my talk with what these findings could mean for African women's reproductive health and birth outcomes. So starting off with a baseline, what did we already know? So I'll start off with the vaginal bacterial communities. The bacterial community exists on the spectrum, and where a woman is on that spectrum is sort of predictive of the possible health consequences that she could have in terms of reproductive health. And on this arm of the spectrum, we have a community dominated by lactobacillus, ranging from L. crispatus, L. gasseri, L. jensina. And this lactobacillus species are actually protective for women's health, and they produce lactic acid in the female genital tract, which leads to a low pH, less than 4.5, and is considered inhibitory inhibitory against diverse pathogens. And so this ecosystem is characterized by low bacterial diversity, and they are also further delineated into what we call community state types, CST1, CST2, and CST5, depending on which bacteria is dominant at that point in time. And then we shift to another arm of the spectrum, where in this arm, the beneficial lactobacillus species are displaced by a diverse mix of non-lactobacillus anaerobes, which are considered pro-inflammatory. They range from Gardnerella, Prevotella, Fanniesia, and so many other diverse species. But when these species are present, the pH is substantially reduced. So it's substantially increased to greater than 4.5. And at that stage, the bacteria diversity is quite high. And this this state is what we call a state of dysbiosis or bacterial vaginosis. In between this spectra, we have a transitionary stage where Lactobacillus iners sits at. So when the female genital tract is dominated by that bacteria, it's considered more of a transitionary stage towards a dysbiotic state. So in terms of the clinical consequences of dysbiosis or BV, is for women with BV, they're more at that increased risk of HIV and STI acquisition, and also they have increased risk for preterm births. And it's estimated that about 50% of African women are affected with BV. So on the other hand, we have the vaginal virome that is comprised of eukaryotic infecting viruses and bacteriophages. with eukaryotic infecting viruses, so many studies have been done on it, quite broad, ranging from studies on human papillomaviruses, anelloviruses, and polyomaviruses. And this is mostly done in the global north, where some studies have characterized their genomes from samples that were taken from the female genital tracts. And there's a growing body of evidence as to their relevance. And then we have bacteriophages. Bacteriophages are the most abundant biological entities on Earth. And in terms of the vaginal virome, they dominate the vaginal virome, but is not quite as well studied as the eukaryotic virome, because there are lots of there's a wide array of diverse phages that till date remain uncharacterized. But a few studies have seen that vaginal lactobacillus species harbor phages in their genomes, and some of them have actually been found from South African women. However, when we look at vaginal virome studies on bacteriophages and we compare that to what global studies have reported, there is sparse genome data from African women, and we remain underrepresented in vaginal virome studies. And this is quite critical, especially because Africa has a high genetic diversity. And I imagine that would also correlate with the female genital tract community. So I'll be focusing mostly on bacteriophages, and I want to take us on a primer of how the bacteriophages life cycle is. So when bacteriophages infect a bacterial host cell, it could take one of two phases. The first one is either the lytic virulence cycle, where the phage infects its genetic material into the bacterial host cell, and then it quickly assembles and replicates. And at some point, it's the bacterial host cell bursts to release the new lytic phages, which could also reinfect other bacteria, and continuous lysis occurs. But on the other hand, it could also go through the temperate cycle or the lysogenic cycle, where the phage infects, but instead of lysine, what it does is that it integrates its phage DNA into the bacterial host. And when the bacterial host cell divides, that gets replicated with the bacterial host cells, but it lies dormant. It's kind of like in the resting phase. But at some point, when the bacterial host cell is under stress, that could trigger that prophage DNA to exit the host genome through a kind of like a SOS response, and then it enters the lytic cycle, where it lyses the host cell and bursts to release new phages. And this is kind of important in the context of reproductive health, as there was a landmark study in the early 2000s where they found that chemicals that are released from smoking can induce prophages in beneficial lactobacillus strains and lyse the host cell. So this figure here shows a lawn of bacteria strains that were grown over a period of, say, 24 to 48 hours. And then they infected the infected induced phages on this bacterial cell. And so these zones of inhibition or zones of lysis, it means that the phages killed the lactobacillus strains. And that was a study done by Pavlova in 2000. And follow-up studies from that also confirmed with electron microscopy the induced prophages from lactobacillus strains. This was actually done in a cohort of South African women. And so over time, there have been opinion pieces as to wondering if bacteriophages could be linked with bacterial vaginosis, in the sense that if these prophages are induced from beneficial lactobacillus strains, is it possible that they could destabilize the vaginal bacterial communities whereby it lyses other beneficial lactobacillus strains, giving rise to an abundance of non-optimal bacteria, and then subsequently leading to BV. So in this image here, from Maderi and Monaco in 2022, they hypothesized or opinionated that in the lactobacillus dominant community, we have those temperate phages resting pretty in the genome. And so there's a low bacterial diversity, low diversity of active phages. But on the other hand, some studies have shown that an increased diversity of bacteriophages is also correlated with increased bacterial diversity, and that is in a BV state. So it's possible that these induced prophages could be actively active in the female genital tract, and also predisposing a woman to increased risk for ST acquisition and transmission. So that is just kind of an opinion. So our first thought was, could clinically relevant compounds that women are exposed to also induce these prophages? And we focus on a few compounds that are commonly used by women in sub-Saharan Africa. The first one is copper IUD. So copper IUD is a very common non-hormonal contraceptive that women use for birth control, but it's been seen in many studies that it causes oxidative stress in the female genital tract and depletes beneficial lactobacillus species. And this has been seen in several studies to be linked to genital inflammation and BV. And so we looked at a wide array of reported pharmacokinetic concentrations in genital tract to take that up for further studies. And then we also looked at tenofovir, which is a popular HIV preventive drug used by a lot of women. So while it's considered safe for use, some recent studies have seen that its protective efficacy has been hampered in the presence of non-lactobacillus dominant communities. So we wonder what tenofovir could be doing in the female genital tract. And so like copper IUD, we looked at a range of published reported concentrations ranging from 0.5 to 8 micrograms per mil. And lastly, looking at hormonal contraceptive, specifically medroxyprogesterone acetate. This is an injectable hormonal contraceptive that is also widely used by some women. While some studies have considered this safe to use, there have been contradictory reports of links to genital inflammation in some cohorts of African women. So the big question is, are these compounds possibly external stressors that are capable of inducing prophages in the cervical-vaginal environment? Please let me drink water. So overall, just to summarize this phase, there's a wide range of there's a great proportion of bacteriophages that are uncharacterized to date. And in terms of studies from African women, we don't have complete phage genomes in public databases, as opposed to studies that have looked at the female genital tract in the global north. So in the public databases, we have tons of vaginal viral genomes that have been deposited, but none from African women. And also, we don't know if these prophages can be induced by clinical compounds and what this could mean for reproductive and sexual health. And so we decided to look at this through metagenomics and in vitro prophage induction assays. So in the next phase, I'm going to be talking about what viral communities exist and what are the possible phage-host interactions that occur. I just want to take you through some of the methods. So we collected vaginal swabs from South African women And then I extracted the DNA from vaginal swab samples, and these samples were sequenced specifically through shotgun metagenomics. And so when we get the shotgun sequences, we get them in the form of raw reads, and we assemble the raw reads into what we call contigs, and group viral sequences into what we call operational taxonomic units. So basically that means that we group them into clusters. So those clusters share 95% similarity. So we group them into those clusters. And then the next phase was to characterize the genomes, identify complete genomes, and what taxonomic group they could belong to. The last phase was to look at predicting the temperate versus lytic lifestyles of the VOTUs, and using a bioinformatics tool, we're also able to look at what possible bacterial hosts those VOTUs could be infecting. So I know my talk is centered mostly on phages, but we also found eukaryotic infecting viruses, complete genomes ranging from anelloviruses, genomoviruses, polyomaviruses. So anelloviruses that are increasingly seen now in the female genital tract, especially in women with vaginitis, women living with HIV, but it's not clear as to what they could be actually doing in the female genital tract for now. Genomoviruses are mostly viruses that infect fungi, and we saw that in the samples we collected, and polyomaviruses. So these lines here basically represent what we call open reading frames in the genomes. So I wouldn't dwell much on this. This is just to identify what genomes are there in the genital tracts. And also with microviruses, we found tons of microviruses in those samples. So microviruses are small single-stranded DNA viruses that infect bacteria. A great percentage of them are considered lytic, and they're widely found in the environment, but many studies have seen them to be found in the female genital tract. It's also not clear what their clinical relevance is as of yet, but they exist. So again, the lines just represent the different open reading frames, but they all vary by the genome length. And this study was published in 2023. If you scan the QR code, it will take you to the paper. So this heat map here represents the phage host interactions that we predicted between Cordovirus viral OTUs and lactobacillus species. So the columns here represents each VOTU. So the numbers are just basically numbers to separate the clusters, but they're all belonging to the same viral class. This here represents the predicted bacterial hosts for these VOTUs. The blue bars are represented by what lifestyles were predicted. So blue bars representing temperate, red bars representing lytic, empty bars representing no predicted lifestyle for that VOTU. So the bars here on the vertical arm, this represents the number of VOTUs that infect that predicted bacterial species. And when we look at the ones on top, this represents the number of bacterial hosts to one VOTU, if that makes sense. So the most distinguishing thing here is that Lactobacillus iners, which is considered a transitionary bacteria towards BV, had the most predicted putative phage host associations to many VOTUs, and it wasn't a shared association with other Lactobacillus species. And again, what was striking was that amongst all of the VOTUs, we find one VOTU that had multiple lytic associations with a wide range of bacterial species. And it's quite interesting because most the earlier studies have always associated a phage to have a specific bacterial host. But a recent study published last year discovered that it's possible that a bacteriophage can infect bacteria from different families. And we wonder if a multi-host bacteriophage interaction could also possibly be happening in the female genital tract. But as of now, this we can't say for sure. Okay. When we look at bacteria that are associated with BV, in this case, Pivotella, there's a broader range of phage host associations that we see, and ranging from temperate to lytic associations. And it's quite interesting that we have more lytic associations than we find with lactobacillus species. And we wonder what could this mean clinically in the female genital tract. So moving on to what drives change. Now that we've established that the phage host interactions, now in terms of clinical compounds, are there other prophage induction triggers in vaginal or crispatus strains? And so for this part of the study, we looked at a study population from the UTUS in Cape Town, South Africa. Yeah, vaginal swab samples were collected from these women, and they were extracted for bacterial DNA and whole genome sequence. So where I came in was to look at those genomes and using certain bioinformatics tools, try to see if there are prophage sequences in these vaginal lactobacillus strains by using prediction tools. So for those that were predicted to harbor prophages, we detect inducible prophages in these strains using a standard phage inducer called mitomycin C, which causes DNA damage and is known to induce phages. So that's more of a control agent. And then having detected those phages, we test for induction with potential stressors, specifically the copper ions, MPA, and tenofovir. And finally, confirm induction via electron microscopy, but also testing the bacterial growth in the presence of these concentration ranges, and sequence the induced phage DNA and do some bioinformatics as well. So prophages for me are always fun to look at under the electron microscope. So we did so at this point, I was working with 4 vaginal bacterial strains, specifically Lactobacillus crispatus. And yes, we confirmed prophages induced with mitomycin C. And so these are phages from the Cordovirus class. They have isocahedral heads and a non-contractile tail. So this is always nice to confirm visually that you have prophages. So having confirmed that, we looked at copper using those reported published concentrations. So this was more of a dose-dependent observation that we saw, whereby the lowest concentration tested, that's this one here, was found to induce prophage similar to that seen with mitomycin C. But as the concentrations increase, we find that there's incomplete phage structures. So just the capsids without the tail, indicating that the copper ions also cause oxidative stress to both the phage and the bacterial host cell. So this shows that the oxidative stress also affected both organisms. But anyway, this is confirmatory that copper can induce prophages. but are bacteriostatic at higher concentrations. So similarly, we looked at the growth curve of the L-crispatous strains with copper ion concentrations. Here, we looked at it over a duration of, sorry, we looked at it over a period of 24 hours. The black here is the uninduced culture control. The blue is the strain that was in the presence of mitomycin C, while the other colors here represents the different copper concentrations. So with increasing copper ion concentrations, we find decreased growth levels in the bacterial strain. And this was the same for this strain as well. So showing that increased copper ion concentrations are also detrimental to the growth of the bacterial host cell. However, we noticed something quite different here with tenofovir. Yes, tenofovir induced prophages at the lowest concentration we tested, which was 0.5 microgram per ml, and which also correlates with the vaginal pharmacokinetic concentration that we see after women have taken oral TDF. So we see that tenofovir induced prophages, but when we look at the range of concentrations to see how the bacterial host cell grows over time in the presence of these concentrations, we find that there isn't much of a change in the concentrations. So the blue here being mitomycin C, and the black somewhere here, which is the uninduced culture control. There isn't much of a difference between the tenofovir concentrations and the uninduced culture. So what could this mean? With Lactobacillus crispatus, it has a unique metabolism of tenofovir such that it actively metabolizes tenofovir to tenofovir diphosphate and then accumulates it intracellularly, thereby reducing bioavailability of the drug itself by 75%. So we wonder if this could actually be affecting the genomes or stressing it to a point where bacteria, where prophages can be induced, but without necessarily hampering the growth of the bacterial cell. But of course, further studies needs to be done on that. So we also sequence genomes of the induced phages, and this we have complete genomes ranging from 44,000 plus base pairs to 48,000 plus base pairs for the different 4 phages. And the different colors here basically reflect their genomic organization as represented by the coding gene regions. And using a tool called Viridic, we also looked at how do our phages compare genomically to reference phages in the genome databases. So all of these viruses in black represent phages that were induced from Lactobacillus species. And it's important to note that most of these phages are from the global north, with none represented by African populations. So the main thing here is that we detected 2 potentially novel species based off of their intergenomic nucleotide similarity. So with this tool, if a phage has less than 95% to its closest reference species, it means that it is potentially novel. And for our species, we had 2 of them having 73.4 and 88.9%. Intergenomic nucleotide similarity to its closest reference phage in the database, showing this just highlights the potential genetic diversity of phages from vaginal strains. And so I'll be shifting to another side of my talk, looking at pregnancy. But here I'm not talking about the vaginal viruses. I'm looking at how the vaginal microbiota changes during pregnancy and postpartum for women living with HIV and without HIV. So globally, HIV, yes, is a pandemic, but in South Africa, we have a national antenatal HIV prevalence of 28 to 30%, which is considered the highest worldwide. And we have the largest population of pregnant women living with HIV compared to other nations. But for women living with HIV, they had increased risk of preterm birth, despite the fact that they are on antiretroviral therapy. And so many studies have actually looked at vaginal bacterial compositions between women with and without HIV, but the key thing is that we don't know for sure what the possible impacts of antiretroviral therapy could be on the vaginal microbiome during pregnancy, independent of HIV. And so for that, we wanted to look at how the microbial composition changes over trimesters and after delivery. So to be able to answer this question, we compared the vaginal microbiota across pregnancy among women with HIV, women not living with HIV, not taking PrEP, women not living with HIV but taking PrEP, and looking at their relation to birth outcomes, specifically preterm versus full-term delivery. And so in this cohort, we had a total of 209 women, 81 women with HIV, 82 not living with HIV, not taking PrEP. For these first two groups of women, we collected samples over a period of between 14 to 36 weeks, second trimester and third trimester, and also at postpartum. However, for PrEP positive women, which was from a different cohort, the samples were collected over the period of 1 month, whereby baseline was the point where they came for the clinic visits, but they were not taking PrEP at that time, but they were given PrEP and followed over the course of 1 month, and vaginal samples were collected afterwards. So just a short-term duration. But the women here were basically scattered across second trimester and third trimester. For eligibility criteria, of course, we look between 14 to 36 weeks of gestational age, looking at babies born preterm or full-term, and excluded women that had gestational diabetes, miscarriages, or experienced stillbirths. So when we did an exploratory analysis to look at the microbiome over pregnancy and postpartum, so here, the brown colors here, represented by Lactobacillus iners, the blue represented by Lactobacillus crispatus, and the different colors here you see are a diverse mix of anaerobes. So we see that for second trimester and third trimester, what clearly stands out here is that the brown colors dominate, which means that Lactobacillus iners dominates during pregnancy. But at postpartum, you see that there's a complete shift whereby Lactobacillus iners is displaced, and we have more of a diverse mix of non-optimal anaerobes. The community state types here, as represented by CST1, CST2, 3, CST4, so these are CST4 subtypes. It depends on what the abundance is at that time. So for a woman having Lactobacillus crispatus abundance, that will be represented by CST1, and CST3 will be represented by this yellow color to differentiate between Lactobacillus iners and crispatus. The Shannon here represents the diversity. Remember I said that for women with BV or women having a dysbiotic state, there's an increased bacterial diversity, and that is reflected here with the diverse mix of non-optimal bacteria, where we see increasing bars So that reflects increasing bacterial diversity. So take home here is that low bacterial diversity, good, high bacterial diversity, bad. So for women with HIV, however, we still see the same trend. There wasn't much of a difference per se as compared to women not living with HIV. But at postpartum, we also see that after delivery, there's a complete shift in what microbiota is there at that moment. We think that the hormonal changes that occur during pregnancy or the protective state is lost. So that affects the vaginal microbiota to the point where there's a delayed microbiome recovery after childbirth. And this has actually been shown in some studies where postpartum recovery takes a very long time, as much as up to a year after delivery. For PrEP positives, also not much of a change per se. So independent of HIV status, there isn't much of a change in the vaginal microbiota during pregnancy, except for postpartum. I just want to point out that postpartum samples were not collected from PrEP positive. So we also looked at to see the longitudinal trend in the alpha diversity over time. So we see that second trimester, third trimester, across groups, there isn't much of a difference, but obviously at postpartum, there's an upward shift towards increased bacterial diversity, but most especially for women living with HIV. So meaning that this is more women with HIV have a higher abundance or a higher diversity of non-optimal bacteria at postpartum compared to women not living with HIV. This also tells a similar story. So this PCA plot represents the different community compositions during pregnancy and at postpartum. So similar to what I've shown you, the CST composition represented by the different types or subtypes, they clearly remain stable from second trimester to third trimester for PrEP negatives. PrEP positives, there isn't much of a change in the community composition. Women with HIV, the same thing. So this is also in agreement with what we have seen with the relative abundance plots. At postpartum, we don't see much of lactobacillus anymore, especially for women with HIV, it's completely lost. But we have CST4, CST3, and CST4 subtypes here. So meaning that community composition remains stable across trimesters. And this was statistically significant across time points. However, when we looked at PrEP positive women to see how the community state types transition between baseline and the visits, off the bat, we don't see much of a change in terms of transitions between community state types at the baseline and afterwards at 1 month. So leading us to say that during this short-term period, PrEP does not impact the vaginal microbiome. We also looked at longitudinal trajectories of the alpha diversity for paired samples, but we don't see a statistically significant difference between those time points. When we stratified the alpha diversity by birth outcome, we see opposing trajectories in terms of women who delivered preterm and women who delivered at full term. So for women who delivered full term, what was interesting was that during the early pregnancy stage, they had increased diversity, which slowly, that's this color here, which slowly, it's kind of stable for women not living with HIV, but for women that delivered preterm in amongst women living with HIV, we see that it's there's a rising shift from early time points to later time points in the gestation period, as opposed to women who delivered at term. So it's possible that the early pregnancy period can be a predictive stage for which women are more likely to deliver preterm. So that was interesting to see. Then we did some further analysis to look at what microorganisms are differentially abundant between women who delivered preterm versus women who delivered term. So looking at differentially abundant bacteria using tools that look at log fold change, but so this is actually separate from relative abundances to see which bacteria are specifically enriched and how significant are they. For women with HIV, the most enriched bacteria is Mobiloncus mulieris. This is interesting because in some recent mechanistic studies, Mobiloncus mulieris has been seen as a significant predictor of preterm births, and that was also enriched with other non-optimal bacterial species, Campylobacter, Lachnocova, Barriella, and none of the good bacteria here for women who were enriched who delivered preterm. However, in the same cohort of women, we find that for women who delivered at term, they had beneficial bacteria present, Lactobacillus crispatus and Lactobacillus jenseni. For PrEP negatives, women not living with HIV, of course, we also had non-optimal bacteria for women who subsequently delivered preterm, and a few beneficial bacteria such as Lactobacillus gasseri, Bifidobacterium breve, and a mix of opportunistic bacteria as well. So similar to what we see with the opposing trajectory plot, second trimester might be a critical window to look at in terms of predicting which women are more likely to deliver preterm, but also a possible window to look at in terms of interventional methods to reduce the risk of preterm births. So big take home here is that Lactobacillus iners remain dominant during pregnancy. PrEP exposure did not significantly alter the vaginal microbiota composition, and second trimester samples from women who later delivered preterm were more frequently enriched with non-optimal bacterial communities. Oh, I forgot to mention that medroxyprogesterone acetate did not induce prophages, so I guess it's safe to use. So we need So after all is said and done, what next? Despite the fact that we have been able to classify some part of the FGT virome, a lot remains to be described and functionally annotated, because when I was analyzing this data, we still find that some viral sequences cannot be characterized specifically because we don't have references for which we can relate them to. So there's a lot to be described, specifically with bacteriophages. And it's possible from what we see in our data that multi-host bacteriophage interactions could occur in the vaginal ecosystem, which means to say that the notion of one phage, one host interaction might not be necessarily what is happening in the vaginal ecosystem. But of course, further studies, we need to look at this. So in the vaginal environment, lactobacillus species do not exist alone. There's a consortium of a wide range of vaginal bacteria, even if lactobacillus is dominating at that point in time. So it's recommended that we further test this in multi-strain consortia cultures to see what clinical compounds concentrations could have when these bacteria are in a mix of other bacterial strains as well. Yes, we see that PrEP didn't have an impact on the vaginal microbiome, but we only looked at a very short window of 1 month. So long-term use of PrEP would provide a more accurate picture on effects of extended exposure on the vaginal microbiota. And from this, insights can be further leveraged into mechanistic studies that aim to improve reproductive health outcomes for African women. So we have only scratched the tip of the iceberg. There's still so much more to uncover about the vaginal microbial ecosystem. There's a lot more to find, and also getting that knowledge to improve reproductive and sexual health. This study would not have been achievable without the study participants, my dear supervisors, the study funders, and collaborators from University of Cape Town, Seattle Children's Research Institute, the study teams, my group members, and the UCT High Performance Computing Facility, Passmore Group, Electron Microscopy Unit. And so this is some of us past and present. You can see Dean hanging over there. So thank you for listening.